the first thematic block, exploring how motivation emotions are regulated in the brains of rodents, non-human primates, and humans. As yesterday, you can ask questions throughout the lectures in the Q&A chat, and we really encourage you to do so. You can also upvote the questions that you find interesting. Now we are pleased to welcome Dr. Ewelina Knapska as our first speaker today. Dr. Knapska obtained her master's degree in biology and psychology from the University of Warsaw. In 2001, she started working with Thomas Werka and Leszek Kaczmarek, studying the heterogeneity of the amygdala and control of positive and negative emotions. After obtaining her PhD from the Nensky Institute of Experimental Biology of the Polish Academy of Science in 2006, Ewelina moved to Ann Arbor, USA for a postdoc in the laboratory of Professor Stephen Maron at the University of Michigan. In 2008, she moved back to Poland and became an assistant professor at the Nensky Institute of Experimental Biology. In 2012, she became an associate professor and the head of the newly created Laboratory of Neurobiology of Emotions. In 2016, Dr. Knapska received a starting grant from the European Research Council to study the amygdala's role in controlling socially transferred emotions. Since 2018, she has been vice president of the Center of Excellence for Neuroplasticity and Brain Disorders in the Nensky Institute of Experimental Biology. Her talk today is titled Secondhand Emotions, What Can Rodents Learn About the World from Others' Bliss and Fear? Dr. Knapska, thanks again for joining us today, and the floor is yours.
Thank you very much. Thank you for the kind introduction and for the invitation. I will try to share my screen right now. Okay, can you see it?
Yes.
Okay, great. So I can start. So it's really a pleasure to be here with you. And I'd like to tell you today about the role of so-called second-hand emotions in adaptation to the environment. In my laboratory, we are interested in second-hand emotions. Second-hand emotions means catching emotions of other individuals. As you know, emotions are typically elicited and expressed in social settings. And of course, they inform us about the emotional state of other individual, a human or an animal, because it's quite common skill. I mean, reading emotions of others in social species. We humans are very good at it, but also animals can read emotions of others. But besides providing information about emotional state of another individual, they also provide us with very important information about our environment. So let's imagine that we meet somebody who is fearful. So of course, there could be different reasons for being fearful, but we can suspect that something wrong is going on in our environment. So understanding emotional expressions of others may provide very important information in terms of warnings against potentially threatening or harmful stimuli. And such learning is very beneficial because we don't need to take a risk of first-hand experience. So we don't need directly about, for example, the presence of the predators in our environment. So we can be more fortunate than these poor creatures here that were just caught by the predators. So if they were warned by their other individuals in their social group, maybe they could avoid this sad fate. And this emotional contagion, and in particular fear contagion, is quite common and very robust phenomenon. And I would like to show you briefly how robust it is. So here is an example of the that such fear contagious emotions. And this situation happened in 2010 during the celebration of War Memorial Day in Amsterdam. There was a big crowd of people, about 10,000 of people, and suddenly a man started to scream. And it was nothing very concrete, no obvious words, but it was just a scream. And this scream caused a panic among the crowd. So you could see it now. So as you see here, it was very, very robust. And this panic resulted in injury of over 60 people. So you can see that humans are really able to efficiently catch emotions of others. And it's, of course, more obvious in the case of negative emotions like fear. And as I mentioned before, animals are also very good at detecting emotions of other individuals. Here is an example of horses, just a family of horses happily grazing. And suddenly one of them detected some danger in the environment. So others followed and ran away. So here is a slow motion movie. So you can see here who started this runaway. So here is a father, a mother, and a child, and a father started to run away, and then the others followed. So such effects, such fear contagion phenomenon can be observed also in rodents, both in rats and mice, and we use it in the laboratory to study the brain mechanisms of this phenomenon. The theory behind emotional contagion is that it increases the probability of similar behavior. So this way it allows for rapid adaptation to environmental challenges. So in the study I'm going to tell you about today, we asked the question whether this socially transfer emotions can provide information about the immediacy of threat. When you think about defensive responses to the threat, they depend very much on the immediacy of the proximity of threat. When the threat is far away, like it's remote, let's say that it's a predator, it's not in the near proximity of an animal, what we can observe are the risk assessment behaviors and increase the vigilance. So the animals are trying to search their environment and localize the source of danger, but they don't stop exploration of the environment. It's quite different when the threat is imminent, like here is a mouse, and I'm not sure whether you can spot it here, but here is also a mouse. So then when the threat is imminent, the animals try to avoid the predator, either freezing. In the rodents, it's a very common response to threat. And this is what we can see here. This poor mouse is trying to hide from the cat here. And probably she's successful because it seems that the cat did not spot her yet. Of course, when it's possible, the animals are trying to escape or even avoid the predators. But in general, the responses are quite different. In the case of the remote threat, when we have this risk assessment behavior, and the imminent threat, when we have either freezing or escape. So we wanted to know whether this different kinds of responses can be evoked by socially transferred information about danger. So to do it, to check it, we used two models we have developed, and we called them imminent threat model and remote threat model. We tested dyads of rats, so the animals housed in pairs, and then one of them was a demonstrator and another was an observer. So in the imminent threat model, we put both animals to the same cage. They were divided, the cage was divided in two parts by perforated partition. And one animal was subjected to fear conditioning. So the witnessing rat, which was a naive animal, could just feel or smell, see or hear what's going on with his partner. So the threat was very imminent from his perspective. In the remote threat model, we used the same experimental procedure in the demonstrator. So it was fear conditioning. We applied 10 foot shocks, so it was very unpleasant. But here in this model, it was in the separate room. So the observer animals couldn't see or smell or hear what's going on with with their partners. So then the partners came back just after the fear conditioning procedure was finished. So they interacted with the observers just after fear conditioning. So these models are can be compared. They are analogous to the situation where an animal was just caught by a predator or just escaped the predator. So we look at the behavior of the observers and compare the observers in those two situations. And we look at the immobility or freezing behavior and rearing behavior. Rearing behavior is standing on the hind paws, just trying to explore the environment. So in this context is risk assessment behavior. So here we have the results from the imminent threat model. And as you can see, the observers froze as the demonstrators did, and they actually froze more than the control animals. And they decreased, both the demonstrators and the observers decreased their exploration rate. The control animals here were the animals that were subjected to the same procedure, but the demonstrators were not fear conditioned, so they were not emotionally aroused. In the remote threat model, we observed quite opposite responses. Was decrease of immobility and increase of exploration of the environment. So here we had this responses that are typical for the imminent threat, and here we had the responses that are typical for the remote threat. So indeed, the animals responded in the way we actually expected. We also measured ultrasonic vocalizations, which are a way of communication between rodents, and we observed the clear differences between these two models. In the imminent threat model, we had mainly vocalizations at 22 kilohertz, which are so-called alarm calls that we can observe when the animals are really fearful. And in the remote threat model, we had higher frequency vocalizations that are typical for less fearful situations. So probably also in this, at the level of this ultrasonic vocalizations, the models differed. And the vocalizations we recorded came from both the demonstrators and the observers. So then we look at the synchronization of the responses between the demonstrators and the observers. So we focus on freezing behavior in the imminent threat model and rearing behavior in the remote threat model, as they were the most robust behaviors in those models. And what we saw was the positive correlation between the level of freezing or rearing between the demonstrators and observers. So the more the demonstrators froze, the more the observers froze. But when we look at synchronization of these behaviors in time, they were not well aligned, they were not well synchronized. So it means that it's not the pure motor mimicry. The observers don't just mimic what the demonstrators do, they just produce similar behavior, but it's not motor mimicry. Then we look at the brain, and we were looking for some neuronal circuits that can be involved in processing social fear. And we focus on the central nucleus of the amygdala. The amygdala is a structure which is crucial for processing of emotions by the brain, both in rodents and in humans. and it's well connected with other parts of the brain. And in particular, the central part of the amygdala is kind of a hub that integrates information from other parts, for example, from the cortex and the basolateral part of the amygdala. And it orchestrates emotional responses by sending projections to different downstream structures. And we focus on the central amygdala because in the literature, it is known that the neuronal circuits in the structure orchestrate active and passive fear responses in case of directly evoked fear. So the fear that is evoked by direct threat like food shocks. So we wanted to see whether in a case of the socially transferred emotions, we also can observe involvement of the structure. So firstly, we look whether the structure is activated in our imminent threat and remote threat models. And we used CFOS as a marker of neuronal activation. CFOS is a protein which is expressed in the nuclei of neurons that are activated. So we can easily measure the level of activation just counting the cells that are activated. So what we observed was that in both models we have some subpopulations of cells that are activated during interaction with an emotionally aroused partner. But of course we didn't know what is the function of these neuronal circuits. So to test for the function of these circuits that are activated, we use the genetic tool we have developed. So the genetic construct that allow for expression of either channel rhodopsin for optogenetic stimulation or Hale rhodopsin for optogenetic inhibition of the neuronal circuits that are activated. They are the expression is under control of CFOS promoter, so we can insert these constructs into the cells that are activated. So the rats were injected with the construct and then subjected to social interaction in either imminent threat model or remote threat model. And then after 24 hours, we tested what happens when we activate or inhibit these circuits, this imminent threat or remote threat cells, when the rats explored the novel environment or interacted with another rat. So firstly, I will show you the results from the exploration of the novel environment. So here we hypothesized that these circuits, the activation or inhibition of the circuits, will modulate the way in which the rats explore the novel environment. So we wanted to see whether it's consistent with this imminent or remote, the defensive responses that are characteristic to the imminent or remote threat. So we used the open field test, which we modified in a way that we added a hiding box in one corner and the opposite corner was brightly lit. So we wanted to measure how eagerly the rats explore the environment, whether they try to avoid exploration by hiding in the box, and whether they are also interested in exploring this slightly threatening area, because the rats are afraid of brightly lit areas. So firstly, we activated or inhibited the neuronal circuits, this imminent threat or remote threat cells in the central amygdala. So firstly, we look at the distance that the animals traveled in this novel environment. And what we saw was that we had quite different responses in when we manipulated with activation of the activity of this imminent threat and remote threat cells. When we inhibited the imminent threat cells, we saw increased exploration of the environment, and we had also increased exploration when we activated the remote threat cells. So it suggested that functionally these neuronal circuits are different. So then we look closer what happens when the animals when we activated these cells, how the animals actually explore the environment. So we look at detailed behaviors related to exploration. And what we observed when we compared the periods when the time was on, so the cells were stimulated, and when the light was off, so the cells were not stimulated, was in the case of the imminent threat cells was decrease of exploration and increase of avoidance. And in case of the remote threat cells, it was increased exploration also of this lighted area of the open field. So here I'd like to show you how it looked like in reality. So here is the movie showing how the rats behaved in the imminent threat model. So now the cells are not stimulated, so the rat is exploring the environment. Also this lighted part. And now we switched on the light, so the rat is trying to hide in the box. So he's showing this avoidance behavior that is characteristic for the imminent threats. Okay, and here we have the remote threat model. The light is off, so the rat is exploring normally the environment. Okay, and now the light is on, so the rat is still exploring even this lighted area, but as you can see, it is cautious. It's like it went to the lighted area, but then turned around and run toward this hiding box. So he's probably cautiously exploring the environment. So, okay, so at this point, we knew that when we manipulate these neuronal circuits in the central amygdala, we can see some modulation of the way in which animals explore novel environment. But the question is whether also the circuits affect social interaction per se. They were activated, in fact, by social interaction. So in the next step, we wanted to see whether they have also some effects on social interaction. And to this end, we used exactly the same method. So we had the CFOS-driven expression of channel rhodopsin, and we stimulated the neuronal circuits of this imminent threat and remote threat cells during social interaction. So the rats were put together to the cage and we stimulated the cells. And what we saw that it's actually the social interaction was not changed. But again, we changed the way of exploration of the environment. So the exploration, the rearing behavior was increased in the remote threat model, and decreased in the imminent threat model. So here is a movie illustrating how it looked like. So the light is off here. So the rat is exploring the environment and his partner. Okay, and now the light is on, so the imminent threat cells are stimulated. And as you can see, the rat is stopped to explore the environment. He's not so interested anymore in exploration. And in the remote threat model, we have quite opposite pattern of results. Now the light is off, so the rats are normally exploring the environment and the partner. Okay, and now the light is on. So you can see that the partner, well, now he's scratching, but now he's rearing. And actually this rearing behavior was the most commonly observed during the stimulation of the imminent remote threat cells in the central amygdala. Okay, so Okay, we knew at this point that the subpopulation of cells in the central amygdala are functionally different. We saw different patterns of behavior when we stimulated those cells. But the question is how they are different. So to characterize them, we look at their activity, we look at their connectivity with other parts of the brain, and we identify the cells that are activated using, again, CFOS expression, and to look at the inputs to the activated, we look at the inputs that are activated in this imminent threat model and remote threat model. And what we found was that there are some differences in the pattern of activated inputs. The difference was in the inputs from the basolateral amygdala and anterior insula. Basolateral amygdala inputs were most active in the imminent threat model, and anterior insula inputs were most active in the remote threat model. There were also some similarities. Anterior cingulate cortex projections were activated in both models. So it seems that there are some similarities, but they are also clearly distinct in the terms of the input projections. We also look at the output projections because, as I said, the central amygdala is a kind of a structure that orchestrates the behavioral and in general emotional responses. So we look at the bunch of structures that are innervated by the central amygdala, and we saw that there are differences in the expression of cells in those structures, and the percentage of cells that receive projections from the central amygdala. So also there are some differences in the pattern of structures that receive projections from the central amygdala cells activated by imminent threat and remote threat. So then we tried to localize or to identify some molecular markers of the cells. As I mentioned in the literature, the neuronal circuits related to active and passive fear have been earlier identified, and they involve two markers, CRF marker and PKC delta marker. So we look at the co-expression of these markers in our models with the activated neurons in the central amygdala. And what we saw was the increased expression of this of CRF, so the about 50–60% of population of the neurons that were activated by the socially derived remote threat were CRF positive, and there was a decrease in the PKC delta cells in this model. So at least partially the neuronal populations that we saw activated in our imminent threat model and remote threat model have similar markers as the cells activated by directly taken threat. So to sum up this part of my talk, I'd like to tell you that rats respond differently to social cues about imminent and remote threat, and it can be explained by the engagement of different populations of the central amygdala neurons. Fear contagion means the change of animals' emotional state rather than just mimicking specific reactions of a conspecific. And we discovered that rats are able to use social cues to flexibly regulate their behavior. So it's possible that if such socially derived cues are so useful in adaptation to the environment, it could stimulate evolution of such abilities, and with time, when they were evolved enough, they could be used as a facility to facilitate social interactions with others. So they can be the basis of more elaborated social skills that are observed in social animals, like empathetic responses. Okay, so here I just showed you the study that we did on dyads of rats. And it's using dyads of rats, it's quite useful in a sense, and quite in a sense easier, because it gives us a good control over experimental conditions. And we are, especially when we are studying the neuronal circuits, the brain mechanisms, we would like to have good control over our experiments. But when we think about social interactions in more naturalistic settings, usually animals, as we humans do, really care who and when who they interact with and when they interact. And actually in our models, when we tested dyads of animals, we decide with whom they interact. We usually randomly put the animals to the cages. So we chose the partners for our rats, and we observed them for a relatively short time. So we don't know much about their social life. We are interested in a very short period of time when we just analyze their behavior. So to focus on the more naturalistic responses, like the more voluntary responses, we built the cage, which we call the Ecohab. The name stands from ecologically relevant habitat. And the cage allows for testing mice living in groups and using larger territory and forming some social structure. So the cage is composed of 4 smaller cages connected with the corridors, equipped with RFID antennas. And every mouse has a transponder with a unique number, and they are recognized by the system when they travel through the corridors of the Ecohab. And the animals are offered food and water here. And in 2 other cages, we have perforated partitions in the corners. And behind these partitions, we can present some social and non-social stimuli. So we can measure how animals are interested in the investigation of such stimuli. The responses of animals, or the activity of the animals, is recorded 24/7. So we can record like large amounts of data for long periods of time, for days or even weeks. We have the dedicated algorithms for analysis of such large amounts of data. And the system in reality looks like this. We keep several such systems in the lab at the moment. We keep them in the individually ventilated cages. So what can we measure with this system? As I mentioned, we can measure approach to social and non-social stimuli. And we usually use social and non-social odors. And we are focused on this olfactory communication because the field studies showed that in the natural conditions, mice actually avoid direct encounters with unfamiliar individuals. It's probably because they can be dangerous. The adult males in mice quite often are aggressive during such encounters. So mice rely mainly on the olfactory communication. So we used in our studies the bedding that is taken from the cages of unfamiliar individuals, or the fresh bedding for comparison, or the bedding from the animals that are somehow stimulated. For example, they are emotionally aroused. So I will show you in a minute an example of such a study. So firstly, we can measure approach to social odor. Then we can measure how much time the animals spend with other animals in the 4 cages of the Ecohab system. So we measure it for pairs of animals, and we called it this measure in cohort sociability. We can also look how animals follow each other in the corridors of the Ecohab system. So as you could see, we have the corridors connecting the cages, and the mice quite often follow closely each other. So we can measure how often they do it. And this behavior is interesting because it correlates well with the dominance structure. So the dominant animals, here we measured it in the classically used YouTube test. So this is the test that is used to establish or to measure social hierarchy in animals. What we saw was the correlation with this following behavior. So the mice that followed others most were the dominant animals. So I will show you now how we can use this different measures in the real study. And I decided to show you the study in which we measured the response to the information about reward in the environment. So we were interested, first of all, whether the animals will respond to this smell, whether they are able to distinguish the smell containing information about the reward from the neutral smell, and then whether they can use it to find the reward in the environment. So in the first part of this experiment, we just put 10 mice to the Ecohab system. Then we separated 2 of them and to single cages, and they were offered either sweetened water or tap water for drinking. The sweetened water meant 10% sucrose solution, which is highly rewarding for mice. Then, after 24 hours, we collected the bedding from both cages and put it to the mice, to the Ecohab, where the other mice still were. And we had this smell signaling a reward here, and the smell signaling that mouse drank just tap water here. So we compared the responses. As a control group, we also had the responses to 2 mice drinking tap water. So when we look at the results, we saw that the mice are indeed interested in this novel smell of this the smell signaling reward. So this is just a urine of a mouse that drank sweetened water, and it's put behind the perforated partition. So the mice have access to the smell, but not to the bedding itself. So what we saw was the increased approach. So they spent more time in this compartment with the reward, and this response was quite persistent. We measured it for 12 hours, and here we compare the response in the second 6-hour period to the first 6-hour period. And we see that when the smell contained information about a reward, this response persisted for the whole recording time. In the control animals, and the animals just exposed to the control smells, with time, this interest decreased. We also observed increased following behavior. So in the tubes of the Ecohab, they followed each other more when this smell of a reward appeared in the environment. So then we wanted to check whether this information can be used in a novel environment to find a reward. So in this experiment, we just put, again, 10 animals to the Ecohab system. Then we transferred 2 of them, so scout mice, to the novel Ecohab system, in which they were offered either sweetened water or tap water for drinking. So they spent their 24 hours drinking from both bottles, and then we removed the mice, removed the bottles, put the bottles with clean bottles with tap water, and transferred the animals from this Ecohab to this new environment, this new Ecohab, and check how they behaved. So what we observed was that they preferred drinking from the bottle where before we had sweet water. Now it was just tap water, so there was no reward in the environment, but they still preferred this bottle, and this response persisted over time. So they showed this preference for 12 hours, and again we showed the increased following behavior. Here we also blocked the plasticity in the prefrontal cortex. Prefrontal cortex is an important structure for processing of social information. And we have the tool that allows for the quite long inhibition of the neuronal plasticity. There are neuroparticles that gradually release TIMP1. TIMP1 is an inhibitor of MMP9, an enzyme which is involved in synaptic plasticity. So we injected these nanoparticles to the brains of our animals, and they blocked the neuroplasticity for several days. So we were able to see how this manipulation changed the behavior. And what we saw is that this effect of the preference of a bottle where the smell of reward was disappeared. And also the persistence was much lower, and the following behavior evoked by the presence of this reward smell in the environment disappeared. So we look at they responded individually to the smell. And here I would like to show you two important things. When we look at this closer at this following behavior, we see that it's very stable over days. And it's like there are clear individual differences. The size of the circle here shows the number of following behavior, the number of following events, and the lines show who is following who. So you can see that there are like super followers, so the mice that follow other more than other animals in the group. They are also mice that almost don't follow other animals. And this is quite stable. So here are the results for 10 days. We observed animals for 10 days. So there are some slight changes, but in general, this social structure is quite stable. So it's important because it also affects how the animals respond to the smell of a reward in the environment. When we put this reward smell to the Ecohab, what we saw was the increase of the following behavior, but this increase was not random. Actually, the super followers increased their behavior most. So because they are also the more dominant animals, it seems that the leaders are actually more interested in what's going on in the environment, because probably this following behavior we observed is the way of searching for the reward in the environment. They are trying to localize the reward. They know that the reward was there before, but there is no reward anymore in the environment, so they are trying to find it. Okay, so at the very end, I'd like to show you our new babies, which is the Ecohab field study, which we did in the field station of my institute, which is in the Mikołajki. This is the region in the northeast part of Poland. It's a very nice area. It's the protected area, so many wild animals there, but we also have the possibility of testing the animals, like laboratory animals outside. So this is what we did there. So we put the Ecohab system, actually several Ecohab systems combined together to one cage. A cage is 2 by 2 meters, and it's divided into 8 compartments, and the compartments are connected with the underground corridors. And each corridor has the RFID antennas, so we can track the animals traveling through the system. And they are quite active, as you can see here. So here is the corridor, and they are using it quite extensively. As you can see, we tested here C57, typical laboratory strain. We offered them food and water, and they are protected from the predators. There is wire mesh around, but they are outside, so they are exposed to the external temperature, which is usually much lower than in the laboratory. We did it in September last year, so it was not very cold, but still it was much colder than usual. They can smell, they can hear the predators, and because this is this protected area, there are a lot of wild animals around. So the environment is much more extreme than in the laboratory. So we hypothesized that in such conditions, we should see the territorial behavior. And we wanted to see whether our observations in the laboratory, which we have with the Ecohab system, are relevant to what we can see under these more challenging natural conditions. So here this is just one result I would like to show you. Here are our networks that are based on the following behavior in the laboratory. So again, the size of the circle shows the number of following events, and the lines show who is following who. The black lines are the data from the Ecohab field experiment. So the solid lines show who is sitting in the same compartment with who. So the preference for spending time together. The dashed black lines show who is avoiding who. So the mice that actually don't like to spend time with others. And here we have plenty of data, so I will try to focus on the most important or most robust connections. So then it's easier to see what actually is happening here. So the mice that are close, they are following each other, so they are probably in close dominance relationships, actually avoid spending time with each other. So they divide the territory between them. So this is in fact related, this dominance behavior is also related to the territorial behavior. But what is also interesting here is that there are mice that are solitaires. So the mice that actually avoid social interactions with others, they don't like to spend time with others. So it's important that there are C57 mice, so they are genetically almost identical, but still we can see the clear social structure and clear individual differences in their sociability that are stable over time. So I think we should take it into consideration when we plan the experiments focus on studying social interactions. So to sum up this part, I'd like to show you that the animals have significant variability in their voluntary social interaction, and that this trait is stable over time and in different environments. We observe that mice respond to social cues signaling reward and can use the cues to find a reward in the environment. And the position in social structure affects responses to social stimuli. And here I'd like to finish and acknowledge input of several people from my laboratory who are behind the data I presented to you today. First of all, Ewelina Knapska and Kacper Kondrakiewicz, who are former PhD students in my laboratory, Karolina Rojek-Sito, Maciek Winiarski, who are current PhD student, and Alicja Puścian, who is a senior postdoc and a person involved in development of the Ecohab system. And also our collaborators from the Technical Warsaw University and Faculty of Physics, who help us in development of this Ecohab system and the data analysis methods, and of course our funding sources. Thank you very much for your attention.
much, Dr. Knapska, for your very interesting talk. It's very fascinating to see how these socially transferred emotions can change the observer's mind's behavior for quite a long time, it seems, especially in this remote threat paradigm. Do you know through which sensory system is most important for transferring these socially transferred emotion, in particular in this remote threat paradigm?
Well, what we studied was the central amygdala and its connectivity with other structures. So this was during the first part. This was the data I showed during the first part of my talk. So this we know. So we know about this CRF positive cells in the central amygdala and some inputs to the cells that seem to be important. But of course, this when we think about the brain, it's probably also other structures. There are probably other structures that are involved in the control of this behavior too.
Thanks. We have a question from the audience. It's from our next speaker, actually, Professor Roberts. Do you see much individual variation in mice responsivity to imminent and distal threat contagion, in the same way that some individuals are more sensitive to their internal physiological responses than others in terms of their impact on the emotional response? I could imagine that some animals may be more sensitive to social external cues than others.
Well, this is a very interesting question. We started to look at it in the Ecohab system. We started with this remote cues, or the cues signaling remote threat, and we see some variability in mice. In the models I showed you at the beginning, it's very difficult to see any variability because we actually try to decrease it by designing our experiments in dyads and in the quite strong stimulation of the animals. When we use in the models in rats I showed you, we used 10 foot shocks. So it's really strong stimulation of the demonstrators. And then the response is probably less variable also in the observers. But when we expose the animals at the olfactory cues, we indeed see some variability. We haven't tested it yet for the imminent threat, but in this remote threat olfactory stimuli, we see the variability.
Thank you very much.
Thanks. The next question is, can you tell anything how the central amygdala neurons behave imminent and remote threat situations if treated with anxiolytic drugs?
Okay, so the question was whether they respond different to the anxiolytic drugs.
Yes, yeah? Exactly, yeah.
Yeah, so we haven't tested it, so I don't know. Well, the anxiolytic drugs usually have the general effects on anxiety and fear, so it may be possible that they decrease the responses in both models, but I don't know it. We haven't tested it.
Thank you. Another question. Thank you for a great talk. Did you study whether activation of the amygdala immediate and remote threat cells can induce social contagion in other rats, i.e. can the emotion be transmitted further?
You mean like the secondary transfer from the observer to another observer?
Yes.
We haven't tested it. Well, it's possible. I think that in rodents, I think it haven't been tested, but when some people are testing group behaviors in, let's say, less advanced, evolutionary advanced animals. So then we can see the transmission of information, also fear, in a big groups of animals. For example, it has been tested in Drosophila, and they are able to transfer information between animals, also in the secondary way. I mean, like there is like a group of animals that are touching each other and are able to pass information about increased concentration of CO2 in the environment. So I think, well, basically such transfer is possible and it would be very adaptive. So yeah, I would expect that it worked this way.
Thank you.
Another question, did you target the lateral or medial part of the central amygdala that is important for the remote or imminent threat?
Yeah, it's for the manipulation studies, when we optogenetically activated or inhibited, we actually manipulated with both parts because it's very difficult to inject the animals just to one part of the central amygdala. In the studies on the connectivity and expression of the molecular markers, we looked separately at both parts because, yeah, there is an important distinction between two. There are some differences, especially in the expression of the markers, but in general, it seems that both parts are involved. We have the increased expression of C4 in both parts, and probably both are involved.
Thank you very much, everyone, for your questions, and thank you, Ewelina, for your very interesting talk. We will have a short break and be back in 5 minutes for our next speaker.
Thank you very much. Thank you for great questions.
Thank you.
It was really a pleasure to be here. Thank you.
Thank you.